Yeah, welcome everybody to I think now our fifth iteration of like the CMFI mass spec seminar. Yeah, it's my very great pleasure to have a guest speaker today here, Pieter Dorrestein from UC San Diego. And yeah, before I'm gonna talk a little bit about the background of the course, just like I want to briefly introduce you, Pieter, because yeah, I'm super excited that he as actually one of like the inventors of molecular networking is giving this lecture today. So yeah, Pieter did his bachelor at Northern Arizona University before he then moved on with his PhD at Cornell University, like all in the realm of like a little bit like structural biology and chemical biology, so investigating like biosynthesis pathways. Before he then joined Neil Kelleher at the University of Illinois in Urbana-Champaign for his postdoc. Relatively short postdoc before he then became faculty at UC San Diego. And yeah, I think what really like, I think is the fascinating part of Pieter's work there is first like the introduction of MALDI imaging for elucidating like chemical exchange between microbial colonies. But then probably more important even, yeah, like the development of like the GMPS infrastructure and in particular molecular networking and yeah, like public data sharing of mainly natural product mass spectrometry data, but now also more and more like metabolomics data in general. And yeah, to me personally, like yeah, reading Pieter's work at the beginning and then being fortunate to join his lab first as a grad student, as a visiting grad student, but then also as a postdoc, this has been really like one of like the game changers for me in natural product mass spectrometry. And I'm still fascinated about molecular networking and how it is developing. And yeah, it's my very great pleasure to have Pieter give this lecture here today. But yeah, before if you're like here for like the first time and you just want to get a little bit background about like the CMFI mass spectrometry seminar. So here again is like the general information with like two important links. First of all, here for like the general information background of the course. And then if you have not signed up already, I would encourage you to do so with like this link here at the bottom of the page. So you can add yourself to our email list. And then, yeah, you can, I don't know, get like date and time changes like today as we actually moved now to the afternoon to better accommodate people from the US. And yeah, so as I said, today we're gonna learn a little bit about molecular networking, and we have a quite a hands-on part where Pieter's gonna walk you through like the job submission of a molecular networking job in GMPS before we then together analyze like the outcome of this. And yeah, without further ado, I'm gonna give over to Pieter, and yeah, thanks again for joining us and doing this lecture today.
Yeah, so I will share my screen and we'll get going. Of course, I'm delighted to be here. So what some of you may already be familiar with GMPS, but not everybody. The way GMPS got started in many ways was a frustration that I had. And that was one of the things that I observed. Oh, by the way, if you have questions, just unmute yourself and ask questions. It's okay to interrupt and feel very comfortable doing so. Or if you're not comfortable unmuting and saying something, just put it in the chat. Daniel, if you can monitor the chat, because when you're presenting, you often don't see it, and just stop me at any time. But one of the reasons why GMPS was established is because I started to realize that I would have students or postdocs working on specific annotations of specific mass spec features. And then I would have a new person come in and they would work in with the same organisms, and they would start over from scratch. There was no dissemination of the knowledge or easy way to take the knowledge from one person to another person. And we thought, well, if we're struggling with this in the context of our own laboratory, then everybody in the world must be struggling with it. And that's sort of the birth of GMPS. Initially, the way I did it in my laboratory, I literally went around with a hard drive getting data from everybody in my lab. And that's really how GMPS started, so that I could get reference spectra of the molecules that we care about. We can actually get the data of people that have collected, and so that we can reuse it from one person to another person. And GMPS is now facilitating this sort of same thought process, but do it at the larger scale so that the community can capture the knowledge of the community in a digital fashion so it can be readily reused by everybody else. So today's sort of objectives is really, if you don't have a GMPS account yet, we're going to create one real quick, and then we're going to start going through creating a molecular networking job. And hopefully, as we start to create molecular networking job, you will start asking me lots of questions. The more questions you ask, the more deeper the understanding will be of what molecular networking actually does. So the first thing that you have to do is go to gnps.ucsd.edu. So that's the website. Let me know if you didn't get there. If you have a username and password, put it in. If you don't have one, register, please. The registration is really simple. You create your login. You need to have one email and then a password, and then go. Do remember what email and password you use or log in, because we don't have a way to reset your password. And so this is your link. And the reason why this is important is because every job analysis that you do is ultimately retained under your own job account. We'll see that in a second. I think there's a question in the chat. Oh, Daniel put gnps.ucsd.edu. Is there anybody that wants to follow along that wasn't able to register or doesn't have an account? If you need me to slow down, let me know as well. Oh, this is what the registration account looks like. So it's username, name, organization, email, password. And again, remember your password and username, because if you do not, we have no way to recreate a password for you, and you would have to use a different email account. And you lose access to all your login information or all your jobs that you've submitted. So then once you're on this page, so the other thing is, if you want to navigate back to the main page in GMPS, you can just usually click the banner. So anywhere in the banner, it will bring you back to this main page. But if you scroll down, what you'll see is this window right here, where you have molecular networking, library search, and molecular blast. Today, we're going to focus on molecular networking. And so click on Create Molecular Network. Is there anybody that wants to follow that was not able to find it? And if so, just put it in the chat or unmute yourself. Okay. So when you hit Create Molecular Network, this window will pop up. And this is the window where we're going to start going, bringing in files into your space. And so when you log in, there's actually a change that happens. So before the login, this is what the banner looks like. So you have massive data sets, documentation, forum, and contact. And so the documentation is the documentation for anything associated with GMPS. So every step-by-step instructions, all the workshops that we've given, there's tutorials, etc. They're all accessible through the documentation. The forum is used if you have questions, you get stuck with something, you want to do advanced analysis and you want to reach out to the community. That's where we use forum. And then of course there is contact information. But when you log in, you can see that this changes a bit. So there's two things that are important, I think, is one of them is the My User Space. So any data set that you upload, any annotations you've made is accessible via My User Space. so that you can see what you've done. But the other one I think is really important is the jobs tab. Every analysis job, so the job that you're going to submit today, will be retained under the jobs tab. I have jobs from 2014 that I can still look at and access and look at the parameters that I set to do the to see how the analysis was done. But also if I want to clone it and use the latest versions of resources that exist in that, you can do that with any link that is under the jobs tab. So this is, to me, this is really exciting. Also, when a job is completed, it will show in the jobs tab, and then you can share the hyperlinks that exist within the jobs tab in papers or with collaborators, so you can start to look at the same job that you have generated. So the first thing that you want to do, once you click on create molecular network and you have this window, is you want to put in a title up there. Make it something that makes sense. So some people put the parameters that they're changing, but for now, just call it Tuesday NP mock mix, or test run of molecular networking, or my first molecular networking job. Name it what you want. So is there anybody who's not able to follow me at this point that wants to follow? Because I'm happy to go back. All right. Now, if you look slightly below the title, so actually the overview window looks like this. Below that, you have the basic options. And so we're going to now, one of the things, yours will be blank here. There are no files selected. What we're going to do is we're now going to click on select input files. And when you do that, the window looks like this, or a window that pops up. Yours might even have fewer data files over here. Some have more if you've already used it. But this is your space on the left. Those are the data files that are already in your space. But what we need to do is we're going to bring in a public data set into your space so that you can use it to do analysis. And the way we do this is that we first click on share files. And when you click on share files, this window will show up. And what we're going to do is we're going to import this massive identification. So massive identification is an accession number to a public data set. And so you're going to type in this number, MSV000-83437, into this import data share.
Before you do that, maybe I think there is a little difference to the actual data set we use. I just pasted it in the chat. It's also the one that's actually written in the field.
Then I am going to change that.
So it's 88759 at the end. For your convenience, yeah, if you just copy and paste it from the chat, that might be the easiest.
Was it
88759.
I thought I had the latest slides for you.
Sorry for that, Peter.
That's okay. No worries. All right, so we're going to redo this. Oh, it was correct in here, just this number was different. So MSV 000-88795, you put it into the import data share. And then when you hit import, it should show up on the right here. Oops. So it should have that X with the MSV 88759. Is there anybody who wants to follow that where it didn't show up on the right here in the import data shares? Okay, in that case, you can then click on select input files. And now you should have your data set on the left side. So now you've imported the public data set, 88759, and you're going to expand the folders. So you're going to click on that plus icon, and this will open up, and then you're going to go to peak. Peak is usually where the MCML files or .mgf files are located. And you will see it expanded, and there are 3 files in there. You're going to highlight those files. You can do it one by one or highlight all 3 of them. And then you can click on Spectrum Files G1. And when you do that, all 3 files should show up under selected Spectrum Files G1. Let me know if sometimes people have they have some challenges in this step where additional folders are grabbed. If that's the case, if there are additional things there, you just highlight it, and then you can click on these X buttons over here, and it will disappear. And so again, just highlight these 3. Hit Spectrum G1 files, and it should appear on the selected Spectrum G1 on the right side. Okay, then hit Finish Selection. And now what you will see in your window is select input files. It should show 3 files and 0 folders. And if that's the case, then great. But if it looks something different, you want to go back and delete the files or whatever you have selected that do not match to those 3. Is there anybody who wants to follow where they weren't able to? So if you don't speak up, I will continue to proceed. Then, because this is high resolution data, we're going to change the precursor ion mass tolerance and the fragment ion mass tolerance. And we're going to change it to 0.01 and 0.01 Dalton. And then check if your email is down below. So it shouldn't be Daniel's email, but it should be your email that you used to create your login account. And this is basically when we submit a job, once it's done, you will get an email there. And because some jobs can take multiple days to run, or weeks sometimes. And so it's kind of nice to have that option. And then you hit Submit. And this window should show up, and sometimes there's some squiggly lines here just showing the progress that is ongoing. Now here, this window I actually really like because it also has clone and clone to latest version. And so what this allows you to do is you can clone it, and you can have, it will show up with the exact same parameters. But let's say you want to change your fragment mass tolerance or some of the other fields, the more advanced options. You can just do that by clone, make the changes, and then hit submit again, and then you have a new submission. And this one will continue to run, but it will it's really easy to redo it. And so here there's a variety of different things that are shown, but basically it shows the spectrum files that you're analyzing, the spectral libraries that you're going to be searching against, et cetera, et cetera. So if you had your own spectral libraries that you added, that would also be listed under here as well. Okay, so now you've actually submitted a molecular networking job. And so of course, you did not know what we submitted, and Daniel will actually tell you the details of what these files were in a little while. But what I'm going to describe to you is the type of job that you actually submitted, and this is classical molecular networking, and it does 2 things. It looks for it matches against reference spectra, so these are spectra of known molecules. But it also looks for similarity among spectra. And so that allows you to build a molecular network. And the way I think about a molecular network is a visualization strategy of data. So it's really a data visualization strategy that helps you. And I will carry you through what I mean by that. So just to give a little bit of background of GMPS itself, when we started in 2010, when we started to have the first molecular networks, which is really the foundation, when we searched against compound libraries, we had less than 2% of the data on average was annotated. I think around 2010, this was probably about 0.5%. And then in 2016, when we actually published GMPS, this was about 1.8%. And so there's basically, if you have some sort of sample, here I describe a human biospecimen just because I give a lot of clinical talks, but it could be an environmental specimen. It could be any specimen, but you do untargeted metabolomics on it. You get this fragmentation data. And I think of these as short signatures that represent molecules. And then you can search against a compound library. So this is usually how we do annotations. And this is the type of annotation approach that is used in molecular networking as well. But then, of course, this was the percent annotation was really, really low. And this is why we introduced molecular networking, because we realized that many molecules, not all molecules, but if they undergo a small modification, that their spectra look very similar. And so if you look for similarity among the spectra, that you should be able to propagate some of these annotations. And that's exactly what we did in molecular networking. This is the first paper where we introduced molecular networking. And so the way molecular networking works is that you have a sample with a mixture of molecules in them. Those molecules have to be ionized. And so, sorry, there's somebody trying to get in. Maybe if you let him in, or I will let him in. So these molecules have to be ionized. In other words, if you have nitrogen-containing molecules, it's really easy to protonate those molecules. And so those are charged. If they have carboxylic acid, it's easy to deprotonate a carboxylic acid so that you can see it in negative ion mode. So once you have these molecules ionized, you can quickly isolate them by mass spectrometry, and then you can subject them to fragmentation. And the way to think about the fragmentation is that you really take an ion that represents a molecule, isolate it inside the instrument, and then you accelerate that ion using different voltages, but you can really selectively accelerate it. And then the chamber in which you accelerate these ions is filled with an inert gas, nitrogen, helium, argon. And then because you have this ion that's accelerating, it now forms collisions with this inert gas. Now, a single collision is likely not going to break up the molecule. But what happens is you convert kinetic energy to vibrational energy during each one of these collisions. And this is important because now you essentially, vibrational energy means you're heating up the molecule. And so once there are sufficient enough collisions, the weakest bonds will start to break apart. And so that's exactly how an MSMS is created. A molecule falls apart into many different pieces. And then you get these fragmentation spectra because you can weigh the pieces that are broken. And so now we get MSMS spectra. We get many of them that represent the detectable composition of the sample. And I should really say detectable because not all molecules are detectable in every experiment. And so you have this pile of MSMS spectra. And then the first thing that we do is we merge all identical spectra. And the reason for this is because that reduces the computational burden to later on compare every spectra to every other spectra based on the similarity metrics. And so there's a tool called MS Cluster with which we do this. We also recently introduced Falcon. There are a couple of different tools that can be used to do the clustering. Within the GMPS environment, the primary one that we use is MS Cluster to do the clustering. And they have different advantages and weaknesses. Some of the clustering algorithms are more greedy, so they will make more mistakes as well. And then others are too relaxed, and it means that you have more spectra of the same molecules that are not clustered. Okay, so now we have MS Cluster, and we merge all identical spectra. And usually what we do is we keep all ions that are found in 60% of the clustered data, or so. That's roughly what we use. The other thing that we do during this process of MS clustering, we actually filter the data. There's two things that we do. First, we apply a square root. What that does, it decreases the high intensity ions a little bit, and it increases the low intensity ions a little bit. And the reason for doing this is because you might, with different instruments, you might have slightly different energies by which they were fragmented. So these intensities may vary a little bit. So we wanted to decrease the contribution to the molecular network later on by decreasing the high intensity ions a little bit and raising the low intensity ions a little bit, but yet that we still retain some intensity information. The other thing that we do is we actually apply a filter. So we keep mass spectrometry data, fragmentation data, always has noise associated with it. And since there's no clear definition of noise, the way we tend to filter is that for every 50 Dalton window, we keep the top 6 ions. And then we start to do MS cluster and compare. And so now we have this these merged spectra. So these are now, we call them unique spectra that hopefully represent unique molecules. But they could also be representing unique ion forms of the molecule. So you may have a sodiated version, a potassiated, and a protonated version. Those would likely represent those would be different nodes, so to speak. So we represent these unique spectra as nodes. And then we do a spectral alignment. And the spectral alignment is basically if you have a parent mass between 2 spectra that are different by a delta mass, we now look for in the MSMS spectra also for those delta masses, and we take that in account when we do the scoring. And then we create a molecular network out of this. And so now we have a node and a delta mass and another node and a delta mass and so on. And so in this case, if the delta mass is, let's say, 2 Dalton, well, that's the difference between a single bond to a double bond. If it's 15.995, it's the difference of an oxygen. If it's 147, that's the delta mass of a phenylalanine. And so this is how you can begin to interpret the information, because if you know this molecule, then you know that this molecule is separated by 147, in other words, separated by a phenylalanine. And so I'm going to show you an example of how we've used, or examples of how we've used molecular networking to really uncover new chemistry or new molecules that exist in biology. One of those was from this particular paper, where we asked a very fundamental question: how much does the microbiome impact the chemistry of the host? And so when we analyzed this, we took germ-free mice, and then mice that were colonized with a normal microbial community. It's called SPF mice, but it stands for specific pathogen-free, which is a normal, healthy microbial community. And we collected untargeted metabolomics on it, much like the one that you just submitted. And we created a molecular network. And I'm just zooming on a very small, tiny bit of the molecular network here. And then we color-coded it based on SPF or germ-free. And what you'll see is that we get this one node that is found in both germ-free mice and SPF mice. we get another node here that is only found in germ-free mice, and as well as here. But then we have 3 nodes that were only found in the mice that were colonized with a normal microbial community. And we searched these masses in the literature, and we could not figure out what they were. Now, we did get a hint, because here we had a spectral match against glycocolic acid, and it's really nicely recognized. So this was a match against the library. What is nice is it's really easy to recognize, because you have this amide bond here, and that fragments quite easily when you do collisionally induced dissociation. And you can see this loss of glycine, and you can actually see glycine itself. And then ions over here actually report largely on this core structure over here, which is, in this case, cholic acid. Now, when you do spectral matches like this, we cannot differentiate necessarily some of the stereochemistry and regiochemistry, even though stereochemistry and regiochemistry are drawn. Mice, for example, also have a molecule called muricholic acid, where this hydroxyl group at the top position is actually localized here. And it's although if you're a true expert, you might be able to recognize some ions that differentiate cholic acid versus muricholic acid. At this point, we cannot yet differentiate it. And so there we took a standard and actually verified it by retention time analysis. But what's important, where I want to get to, is that this was a handle to understand what some of these other signals are that we see in this particular network. And that is because here we see a 90 Dalton difference. So we go from 466 to 556. Now, if you've ever done peptide analysis and mutation analysis in peptides, you might recognize that this is a difference between glycine and phenylalanine. And so our prediction was that this was a substitution of glycine to phenylalanine. And that's exactly what we saw. If you inspect the MSMS data, we see this loss of phenylalanine, and here we see the phenylalanine signal, which is great. Then here we see a 15.996 Dalton. And if you look at the periodic chart, you can actually figure out what this is. But let's say you did not know what this signal was, or what this delta mass may represent. There are some resources, and there are 2 resources that we now use. One of those is Unimod. Unimod are actually post-translational modifications of peptides. Now, a lot of the modifications that people see on peptides are also often found in small molecules. And so this is a really wonderful resource, and you just have to put in the 15.99, and then what you will see, okay, we see a variety of different things, deoxy, oxidation, and the composition is always oxygen. And so what that means is that it's likely some phenylalanine, because we determined that this was phenylalanine conjugate, but that it has some oxygen attached to it. And so when we look at it, indeed, we see that it's a tyrosine modified version. And so here's the tyrosine, and you can see the tyrosine again. And so this is one way that you can propagate some of these annotations. So now I'm going to ask you real quick. So here we see a 79.993 Dalton difference. If you use these resources, such as Unimod, or this Google spreadsheet, so this is a community curated spreadsheet, we can't vouch for accuracy. Can you tell me what these 79.993 might be? And I'll let you do that for a few minutes, and just post it either in the chat or call it out after unmuting yourself. So the other question is, did I go too fast? Do they have the link to the slides?
Not to the slides.
Okay, can you post the link maybe?
Oh, so we have the example link for the other network because we submitted the tubing and natural product mix.
Correct, but I'm trying to get people to tell me what they think 79.993 is.
Okay, so which link do you want me to share?
Probably Unimod would be good.
Oh, okay.
So for Unimod, there's a guest login. Yeah, Keshab. It's phosphorylation or sulfation. That is absolutely correct. If you look at the very high resolution data, you will actually figure out that this is sulfation. And so in other words, in germ-free mice, we see some of these sulfated glycocolic acids that are not observed in the mice that are colonized with microbes. And so this is how you can start to begin to get additional structural insight. And so what I'm going to do next is I'm going to carry you through 2 more examples. I won't ask you questions, and then I will let Daniel take over. So the next example I want to show you is the indexing of Pseudomonas. So these are a large number of Pseudomonas strains that were analyzed by untargeted metabolomics. We build a molecular network shown right here. And we're going to focus on this particular molecular family up in the upper left-hand corner. And so here's the family itself. And when you inspect some of these spectra that you see right here in circles, what you will recognize is that these end up being peptides. And that's because the delta masses match up to the delta masses associated with different amino acids. And so what you can then see is that this red one, if you go from the red one to the blue one, that they are separated from valine to isoleucine leucine. And then if you go to the green one, now it's an insertion of a leucine. If you go from the green one to this yellow one, you can start to see that it has a lot different substitution. So an E to D, S to Q, valine. And the way we determine that is we simply look at the delta masses that we see in these peptides. And then we have one that is still related, but is going to be very different. And so because we have these libraries of known molecules, we can do dereplication. And so you can begin to annotate some things. And so here, the red one happened to be viscosin or lip. The difference between these two are the stereochemistry of one of the amino acids, but overall structure is identical. And then adjacent to this, we had viscosamide. You can see the only substitution is a single amino acid. And it's really the hydroxyl group versus the ammonium. And so again, this was dereplicated to viscosamide. This one was macetonolide. Again, single amino acid substitution. And keep in mind, each of these molecules are found in different Pseudomonas strains. So what you're really seeing is evolution in action, so to speak, from one organism to the other organism, where they change the biosynthesis just slightly to make new molecules. And then here we have an insertion of an amino acid. And then here the fatty acid is changed. We have another amino acid substitution. And then over here we have dereplicated to tensin. And then this one was an unknown. And based on the analysis of the fragmentation data, we can predict that the changes were happening at this fatty acid position, as well as these two amino acids. This is actually 2016 paper. So then the next example is from an actinomycete, and or a marine microbial data set, I should say. And so we created a molecular network from about 1,000 LCMS files. And so you can now overview all the data that exists in that large data set that we've collected. And we dereplicated all the ones that are annotated here. So these are known natural products. But a lot of people in the natural product community care about molecules that are unknown. And so here we had a cluster, for example, that did not have an annotation, and we're going to zoom in on this. And what you will see is that molecules with a mass of 900 cluster together with molecules that have masses of 1,300. And so in the past, people would have said, well, they cannot be related to each other because the masses are so different. And so we did the structure elucidation for 2 of these molecules, and now it will make sense how they are structure related. It was basically a polymeric substance that is being biosynthesized by this particular microbe. And you can see that we have a mass of 972 versus 1360, and that's basically all the masses that you see in these clusters are all separated by these different units shown right here. And so this is how you can use molecular networking to interpret and discover new molecules. And at this point, I'll let Daniel take over. And if there are any questions, do you have questions in terms of how networks are computed? How, I mean, now is your opportunity to ask questions.
All right, if not, we can also have a more informal discussion afterwards, which won't be recorded. So in case you're shy and you don't want your questions to end up on YouTube. Okay, but yeah, before we get there, I want to use like the last 5 minutes with you to just quickly explore actually the data that you just submitted to like that molecular networking job with Pieter, and like, yeah, perhaps find already new molecules yourself. So yeah, so the last objectives are simply like exploring the molecular network in the web browser, and then yeah, like find analogs of some known natural products. And this is not only like a task for now, so also maybe later on, if you want to like click a little bit around and find new molecules, then I think that data set we just submitted for molecular networking would be perfectly suited. So now a question, did you guys get emails from your jobs that were finished? So who got an email from GMPS that the job you submitted finished? If you quickly check it. Okay. All right, then unfortunately, we are just probably above like the limit of like processing time to make it like on time for exploring your data set. But I have a backup link here, which I can also quickly share with you in the chat, which is basically exactly what you did. And I think this is also the nice example of yeah, how easy and comfortable it is to share your results through like such an URL. So this is literally just like once the job was done, I copied the URL out of my browser and shared it now with you. And now when I actually click on this, then my web browser should open, and you should also see this here through screen share. Good. Okay, then yeah, basically the same yeah, you should then have also in your own web browser when you clicked on it. Maybe if I can just get some thumbs up that this worked. Awesome. And I think, yeah, that's already like the result of your molecular network. And also here, what Pieter highlighted, if you want to modify some settings, there's also like this clone buttons here. And especially if it's been like a while since you run that job, I would recommend to use like clone to the latest version. That way you also make sure to run it like with the latest GMPS version. And yeah, now to just like jump right into like the molecular network, there's basically a couple of ways to get there, but like the most straightforward one is probably click here on this view all clusters with IDs. If you basically click on that, what it will show you is a list of all nodes in your network. So we call them cluster because those are basically clustered identical MS spectra through that MS cluster algorithm. So you have to get familiar a little bit to like the different like nomenclature of the GMPS things, but yeah, cluster in that.
This is a clash of two different fields coming together, because you have the molecular network, which the network a lot of fields will call clusters, and then here in this case clustering means all the identical spectra that are merged. So both terms will be used, but they mean slightly different things. And that's because the systems biology community that created the networking infrastructure to be enabled to visualize the networks called it clusters. And then the computer scientists where we build MS cluster with, they call it the merge spectra clusters. And so it's just sometimes when you have two disciplines coming together, and it's actually quite common, they will use the same terms for different things. So just be aware of that.
Yeah, so I think the way I try to remember this is by remembering MS cluster. So that's the algorithm that merges like the identical spectra. And especially if you have like many files that all hit the same molecule, then you need like that to like reduce redundancy. And yeah, this is called MS cluster, and obviously MS cluster generates clusters, and then those clusters are connected in the network. And yeah, here in like view all clusters with ID, we basically now get this list of all the nodes or clusters of our network, right? And when you like look here on the top left, you would see that there's a total of 1,358 clusters. So that's basically our global network. So right now in the browser, I can't really like visualize this as a whole. Therefore, I would need to like actually download this data and put it into like Cytoscape. But we can go through like that list and look at like individual molecular families. So those are so-called subnetworks. And I think for our task now, finding new analogs, this will just be perfectly enough, because there's a nice way here to filter, for example, like by library ID here on the right. So if you click on that, then yeah, it will basically sort now all the clusters based on their library ID, and you can see already, oh wow, there's a bunch of compounds, and depending on your chemical, natural product, chemistry background, maybe some of them sound exciting. If not, you can just like click here on the next page, and then we see some more. Okay, yeah, there's some tubing specific compounds within those. So I don't know, maybe you find one. Anyway, now when I look at those particular compounds and go to like the left, then I see eventually here under spectral family that there is a view network link. And that is basically like the one click away to the molecular network of those. So for example, here for like rapamycin, a known natural product, I can click on it, and then, oops, what happened? View network. Oh yeah, probably clicked the wrong link. So then, yeah, basically like this network visualization tool here should appear. And then I see, oh, this network is actually just 2 nodes here. And when I click on like the parent mass, then I would see, oh, okay, so but they have at least different precursor masses. And when I click here on edge label delta MZ, then you would see that there is a difference of 14. So now I can also click on the node itself. So it will display me here like the mass spectrum. And now when I right click on the other node, it will display the other mass spectrum here below. And then I can here use this magic stick to also annotate the masses. But yeah, probably more important, I also see like here the structure, and then I have like the delta mass of 14.015. And yeah, maybe like some of like the chemists or natural product enthusiasts among us already have an idea what this delta mass could refer to. I don't know, somebody wanna quickly say it? I see Raphael is ready. 14. It's probably one of like the most frequent delta masses we observe in molecular networks. Yeah, it's typically like CH2. So like either like a methyl group, or especially if you have a peptide and like some aliphatic amino acids, then it's often like a substitution between, I don't know, glycine, alanine, and so on. Anyway, so now one of those, the blue one, was annotated as rapamycin, but the other one did not yield any hit in the library. So this is already a potentially new derivative, like at least according to our spectral knowledge in GMPS, against which we dereplicated this data. So now, I don't know, we can go on with that thing and yeah, like maybe try and find something with a larger network. So I'm just going to go back to my status page here and then view all clusters with IDs again. And then, okay, now I sort and well, anybody has a favorite? I just pick one. Also just a duplet. Oh yeah. All right, at least 3. Okay, so now here, interesting. Okay. So now we have an interesting case here, because actually 2 of them would have the same precursor mass, right? So now I can inspect a little bit like the mass spectrum. And now that could be like 2 things. Either it could be like an isomer, where perhaps like some of like the like residues have like a different location, but it could also be an artifact that actually it was an MS2 spectra that was, I don't know, like of lower quality and was not like really like hit by MS cluster and merged into like the original cluster. So, but then there's also another neighboring one that indeed has a different precursor mass. Okay, 15.972. It's also an interesting one. Somebody knows what that is? I'm sure that there are like a bunch of you know what that is. Come on.
And if you don't, you can go to the Unimod website or the other link. You can type in 15.9 and you would find it.
Yes. Is it CH4 or oxygen? No.
No, because when you look at like this very exact mass, then you see that it's a little bit below the nominal mass. So that means that it has a negative mass defect. And
yes,
yeah, like,
but it's a really good guess, because 15. CH4 is 16 daltons.
Exactly.
And so it's absolutely correct because you have 2 protons and in addition to the CH2. So CH2 tends to be methylation, and then you would have to have another 2 Daltons. But CH2 is 2.015. CH2 is 14.0, I think also 15. And so if you add those together, it should be 16.03 something. And that would be CH4. But this is 15.972. And so this is what Daniel is talking about, the mass defect, which is the small masses at the end that really indicates that would not be correct. And so if you type in 15.9 in Unimod, what you will find out is that this often matches to oxygen. Well, it matches to oxygen. And so, as Daniel was saying, the nominal mass was in agreement with your guess. But if you start to look at the mass defect, it's not correct. Does that make sense? Was that helpful, the explanation?
Yeah, thank you, Pieter. Upser? All good on that? Yeah, so it's really like the tiny differences in like the 3 or 4 digits here that like give you like a very good like hint that this is most likely like oxygen. And then for like the other masses, it's the same. I mean, it's a good way of thinking. Like first think about like the nominal mass, but then yeah, with like high res data, it's really like those mass defects that that point you towards a certain direction. And yeah, oxygen is one of the few elements we typically deal with that puts it like to the more like negative side towards like the nominal mass. So yeah, so here we
so there was one guess of NH2. NH2 has a nominal mass of 17, and so it's slightly off. There are occasionally that you will get a substitution of a proton with NH2 that it may appear that it's 16, but it would still have a different mass defect than oxygen.
Yeah. So yeah, I think in this case, I think the most likely explanation would be indeed then here a difference of an oxygen. So this could be an oxidated version of cyclosporine. And now when you understand the fragmentation behavior of it, we're not going to go into that much detail, but actually by inspecting, for example, like the B and Y ion series, you could actually narrow it down to a specific site of your molecule. And then, yeah, according to this mass difference, like propose like a new structure. And yeah, I think with that, I want to leave this to you and encourage you to take a look at it, and maybe you find some. And yeah, if you, I don't know, next time we can spend the first 5 minutes or so, and if you want to showcase one, that'd be awesome. So yeah, with that,
for maybe before you go on, just to say, rapamycin, cyclosporin are really important molecules. They are actually used in the clinic to prevent organ rejection during transplants. And so that's really been enabled by molecules such as rapamycin and such. So it's a really unique data set to look at.
Yes, so yeah, I don't know, maybe you find some other germs. So gems within that data set. So yeah, it's a mock mix of different natural products we had here, and hopefully over the next couple of weeks we will also explore the space a little bit more of like the, I don't know, wealth on compounds that also have been discovered here in Tübingen. So I'm looking forward to that. And yeah, if you want to, I don't know, like help us like finding new molecules in there, that'd be pretty awesome. But yeah, I think for now, I just want to leave you with that link again if you haven't like opened it yet. I'll also put it in the in our document and in the video caption later. And then yeah, just like a little preview for what we're gonna do next time. So next time we will have Ming Wang from also UC San Diego showcasing the GMPS dashboard to us. So how we can now go from like this molecular networks actually back to the raw data. And then one very particular cool thing is Ming will highlight the use of MAST to search then new spectra, which we eventually discovered against a full repository, and check how where else like in different environments those compounds might be found. So yeah, I'm very much looking forward for that. And yeah, thanks again for joining us, and especially thank you, Pieter, for giving the lecture today. It was a lot of fun to listen to you. And yeah, now I think we have some time for a more informal discussion. So if you want to stay on and ask Pieter your questions, I'm sure he would be happy to answer them. So yeah, thanks again, and hope to see you next time.